going to get started, and the first thing I want to say is that this is informal, so I would like for you to ask questions whenever you feel you need to. It's okay to interrupt. If you don't want to interrupt verbally, you can type something in the chat box, and Deb will let me know when there's questions. But either way, it will probably be easier for you to, for us to answer the questions if you just go ahead and ask them as they come up. So I'm going to start on the Hymenoptera Genome Database homepage, which is where you can enter HymenopteraMine. And I want to point out for people who have used Hymenoptera Genome Database or BBase or Nasonia Base or the Angenomes portal in the past, that the whole website has been updated and reorganized. And so there's information about the reorganization here under the HGD updates. So the most important thing, I think, for the people who've been using this in the past is that the older websites are available in this archive section. And this is just mainly so that you can download the older data sets. So, so you can still, those will be there and always there, but they won't be updated any longer. So currently all of our data will be now in this unified Hymenoptera Genome Database website, and that will make it a lot easier for us to incorporate more genomes in the future. And so now you can see that all the current downloads are available with this one tab. For example, if you click on genome files, there's all the FASTA files that are used in our, the genomes used in our genome browsers and in HymenopteraMine. And also this genome publications tab has the publications for all the genomes that we have. And in some cases, these have not been published. And so when they aren't published, it will tell you. And then, for example, we have a genome in HymenopteraMine that's an I5K pilot project. And so there's a link provided to go to I5K to find out about the data sharing information. So
I had an opening question about the data.
Yes.
Will data in HymenopteraMine differ from data in NCBI?
Well, it will, because we can load HymenopteraMine once a year. It's a big job for preparing a new load and integrating this data. So if, let's say we complete our next release by next fall, then a new genome, a genome upgrade that happens in December won't be in our new release, in our latest release until the following year. So it's not, we can't do live updating as new data becomes available. It's a kind of, we have a data freeze at a certain date, and then it takes us a couple months from that freeze to actually process all the data and do the load. So it will, as a result, it could be a step behind what you'd find at NCBI. And so it's a good idea to check. And so I'll show you in our data sources page how you can find out what assembly we have. And so I had an advisory meeting last fall, and it was requested that we do a better job at providing the dates for our data. So we will try to do that from now on. If we go to the home page, it does mention, that the data that we currently have is from a data freeze of April 4th, 2019. And so our next release, we plan to base that on a freeze that we had in May. Although, we might be able to squeeze in a few more genomes that become available maybe in the next month, but we'll check NCBI again just to make sure before we do the next load. But it always takes a few months to load after we do a freeze, and so we'll try to always provide the date here. We have a new tab called Contributing Data. So there's no way for anybody to actually upload data to Hymenoptera Genome Database. It needs to be just basically communicating with us, and if you have data that we can incorporate, then we would work out a way to get that from you. So we would either provide a way for you to upload data to the server, or there'd be some other way. So for getting data into HymenopteraMine or Hymenoptera Genome Database in general, we prefer for there to be a RefSeq genome assembly and gene set, because by being in RefSeq, it allows the genes to be connected to many other data sources such as KEGG and OrthoDB. If it's not in RefSeq, then there's a good chance that even though there might be genes in HymenopteraMine, it might not be those genes might not be connected to other kinds of information. Let's see. One thing I wanted to say about this genome publications page is that when you use Hymenoptera Genome Database or HymenopteraMine, of course we want you to cite the Hymenoptera Genome Database paper, but it's also very important to cite the genomes that you use. And so we try to keep this page updated for all the genomes, and every genome that you use in your analysis should be cited in your paper. So I'm going to go back home. Something else I'd like to point out about our homepage is that it mentions that we have a chapter in Methods in Molecular Biology, a volume called Eukaryotic Genome Databases. And this chapter has some very detailed step-by-step examples of HymenopteraMine. So you can either download it from Methods in Molecular Biology using this link. However, that is not free access, so some people might not have access to it. And if that's the case, you can download a free preprint from our website, and that has all the same content. It's just not as formatted as nicely. Although I would say that the figures in the preprint are probably easier to view than the figures in the published version, because the preprint version has the original PDF, so that might even be useful even if you can get access. So this chapter was published a couple years ago when we were on a previous release of HymenopteraMine. So if you go to this HymenopteraMine tab, you'll see that we actually have 3 versions of HymenopteraMine available. And so the chapter, Methods in Molecular Biology chapter, if you want to do the step-by-step examples and have it look exactly the same, you should use this oldest release. And something else I'd like to say about the releases is that you need to register. I'm going to tell you about registering for using HymenopteraMine. It's not necessary, but it increases what you can do. But if you do want to register, you would have to register for each one separately if you want to use different versions of HymenopteraMine, because the registration is connected to each one separately. So our latest version is 1.4, but we leave 1.3 up there because of data changes. So some of the species that were previously in OrthoDB 9 are no longer in OrthoDB 10. So people who are using those species might actually want to use the previous version of HymenopteraMine. Okay, so now I'm going to actually click on the HymenopteraMine link to go to HymenopteraMine. So each one of those links will look almost the same. It will look a lot like this, except for there'll be a different version number, and some of the content might be different. But here, for today, we're going to look at the latest release. So the first thing I want to tell you is how to create an account. So an account is not necessary for many of the kind of just quick searching that you might do, but in order to do some of the more, complex searches, like saving lists of IDs and being able to do searches with those lists, you need to actually have an account so you can save the list in your account. So if you click login, you have both a login area, but you also have the create account area. So you would click on create account now if you don't have an account yet, or if you want to use a different version that you don't have an account on. And all you need to enter is your email and make up a password, so it's very easy. So I'm going to actually go ahead and log in now, and I have several accounts. This is my demonstration user. So now that I'm logged in, I'll show you some of the things that you can have when you're logged in. So you have this My Mind tab that allows you to look at different things. So here are some different lists that I've saved in my account. I can look at a history of queries that I've done. I only set this account up yesterday, so I don't have very many queries in here yet. If I actually create new queries, then I would have them listed here, but I didn't, so I don't have any here. There's a place where you can change your password. I'll tell you about templates later. And there's also this account details that allows you to get an API key if you want to connect to HymenopteraMine programmatically. It allows you to change your public username if you want to share your list with other people using a different username, and update your email address. Yes? Is that a question? Okay, no.
We have one in the chat though. Are we able or encouraged or discouraged from creating more than one account?
It would be fine to create more than one account. I think it would be nice. So sometime in the future, we would like to be able to contact users so that we can let them know when we're going to update HymenopteraMine. And so I guess you might receive 2 emails if you create 2 accounts, if you have using different email addresses. But for me, I personally have more than one account because I have an account that I use in my research that has a lot of lists in it that would make things confusing when I'm doing tutorials. And then I have my, my really clean looking account when I'm doing a live tutorial, and I have my other account for when I'm looking for examples. So, but it's fine if you have more than one account. Okay, so now I'm going to go back home. So I'm going to show you a little bit more of what we have on this home page. So first of all, there's if you click on this help tab, you'll get to these read the docs documentation. So this has a lot of information on how to use the different tools, and then also has a link to our YouTube channel. Let's see, let me go back. Then we also have these tabs are for different tools that I'm going to be showing you. And then we also have this data sources page, which this is an important page because this lets you know which versions of assemblies you have and which gene sets you have. So for some of the species, some of the older species, I would say, we have the official gene sets that were published with their original publications. But those, pretty much almost all of those are old now. Most of those are from older assemblies, and they've had to be mapped to new assemblies. So they're probably not the best thing if you're starting a new research project to actually be using one of those. So you should always check to find out what is the latest assembly before you start a new research project. But we like to provide these because sometimes you want to compare your current work with what happened and what was published. And so this will allow you to do cross references between the new genes and the old genes. So we have, these are the species that have official gene sets and which gene sets that we have. We also have RefSeq genes for most species. So there's a few RefSeq genes species that don't have RefSeq genes, and for historical reasons, we are keeping those in here. I think we only have 2 of those species, but in the future, as we add more, we want to focus on the RefSeq genes because those are the genes that are connected to other data sources. And so this tells exactly which assembly those genes came from. So we have homology. And so you'll notice when I show you some of these other data sets that not every species is listed, and that's because these other data sources don't have every species. So if you're using it for your species, you should look up to see, if I want to look at OrthoDB, is my species in this list. And another point about OrthoDB is that the newest version of OrthoDB tries to keep up with the newest assemblies, and so therefore they tend to use RefSeq gene IDs rather than official gene sets as well. So we also have protein information from UniPro, protein and family and domain information from InterPro, gene ontology annotation, although I have to say that we currently get gene ontology from UniPro go away, and some species have very low representation in that data set. So even though it might be listed here, you might find out that the number of genes that are annotated with Go for your species is too small for you to actually do a good gene ontology enrichment. So we've developed a new gene ontology annotation pipeline, and we'll be running it for all the species. And so all the species will have gene ontology annotation in the next release. So right now I just say if your species is not Apis or Bombus or one of the older older genomes that was published long ago, then you might not have great representation in gene ontology currently. And then here we list all the different genome assemblies. And then we have publications from PubMed. We have pathway information, KEGG for some of the species. These are all connected to the RefSeq gene sets. We have pathway information from Reactome for Drosophila melanogaster, and Drosophila melanogaster is the one species we have that is not Hymenoptera. And the reason why we have that is that it allows you to identify orthologs using the OrthoDB information and then maybe connect your information with Drosophila information. We also have interactions that were available only for Drosophila or Drosophila and Apis mellifera. And then we have a couple ontologies, including, of course, gene ontology. So this is an important page to always look at when you're working with HymenopteraMine to find out what is available for your species. So we'll go back to the home page. So now I'm going to start showing you the tools on the home page. You'll notice that we have these little boxes up here, and then we have this bar that goes across in the middle. And so what this bar is it's a bar that divides up these things that we call query templates, which are pre-built queries, and they're organized into the kind of data that they have. So if you just click on each one, you'll find out there's different kind of these queries that are listed, and I'll go over these pretty soon. But the first thing I want to do is actually go back up to the easiest way to search, which is with the quick search box. So the quick search is basically something that does a text lookup of all the records in HymenopteraMine to see if it can find that text term. So if I just, for example, let's say I know part of a gene symbol, but I don't know the whole thing, I can just enter, I can use a wildcard, an asterisk as a wildcard, and do a search with that. And so after you do a search, it tells you what kind of records it found that in, and then also which organisms. And so, so you can see these are some different genes, and let's say I wanted to find all the ARP genes, then just by using the wildcard, I was able to pick up some different ones. Let's say I just want to look at the Apis genes or Apis, then I can see Apis has it only in these records. It doesn't have in publications. And I want to actually just look at the gene, so I can click on that, and then here it is. And so if you click here, you'll get to the report page for that gene. And this report page has typical information that a gene report would have, but the main difference is that it's organized into tables. So and these tables are each downloadable separately. And I'll show you more about tables later, but just to show you some of the things that we have in the gene report page. One thing that we have for some of the species is these things called alias and database cross-references. And so a database cross-reference means that we have two gene sets in HymenopteraMine, and we can cross-reference each other. So this is for those older species that had official gene sets. So if I look at here, I can click on this to open up this little window for database cross-reference, and you'll see that this NCBI ID cross-references this old gene ID from email OGSV 3.2. We also have some species have alias names, and so in this case, these might be from very old gene sets, and these genes are not loaded into HymenopteraMine. It's just that the information, their IDs have been attached to the genes that they're most similar to, that they map to. But I can't click on a gene on this ID and go to another gene report because this gene is not here. It just allows you to connect your new genes with these very old genes. Whereas if I clicked on this GB ID, I'd actually go to another gene report page. So that's the difference between alias and database cross-reference. Database cross-reference tends to have the full information. So we have transcripts and the mappings of the the protein IDs to transcript IDs and proteins from UniPro. So here's the UniPro ID. Gene ontology, this one doesn't happen to have very many terms. Pathways, homology, which is available in 2 different formats. And so these tables are, I'll show you later, but you can export each one of these separately. Publications. I'll just go back to the home page. So that was the easiest way of searching HymenopteraMine, but it's probably it's maybe good for kind of exploring to see if what you're looking for is there, but it's probably not the most useful way. More likely you want some kind of more complex query. So the next level of complexity would be to use one of these templates. And so I'm gonna just click on this function category and show you what a query template looks like. So just this one is called gene ID to pathways. And so what you can probably guess is that you enter a gene ID and be able to get pathway information. So I'm going to just leave the default ID in here. So all of these query templates will have a default identifier that you can change, but I'll just leave it just to make it simple. Click show results, and then there are the results for that query. So if I want to go back to that template really easily, instead of going to the home page, I could just click on query up here. And then here I'm back again. So now I would like to actually edit this query, and I'm going to show you a little bit of the query builder, but I won't be showing that much of it today because it's I'll save that for a more advanced tutorial. But if I click on edit query.
Would you mind going back to where you got there, please?
Oh, yes. Okay, so I'm, let me just, starting from here or let me just I'll do the whole thing again. So I'm gonna go back to the home page, and I was interested in pathways. And so pathways would be something that would be under a function.
Okay.
And then, so I clicked on this function tab, and then there's several different kinds of queries that I can do here. And there's actually even more if I click more queries. But just the gene ID to pathways is the one I showed you. So I'm going to just click on that, and then it opens up this menu. And so the first thing I did was I didn't change anything at all. I just said show results. So when I do show results, these are the results. This is actually a pretty small table because it's the pathways for one gene. But this is, I'll show you a little bit more about the tables later, but these are the results of that query. But let's say I actually, I see, oh, this is a good query. I want to do this query, but I want to change it a little bit. I can go back to here, up here. Okay, on the upper left side, see my mouth. You know, it's probably hard for you guys to see my mouse moving sometimes, but up here where it says trail query, I click on query, and it just goes back to the same template, so you don't have to go to the home page again. And so now, if I would like to change this query, I can click edit query, and I get to the query builder. This is the tool that we use when we're building the queries, and if anybody ever has any problems changing their query, they can just click the contact us button up here, and we'll help you with it. We'll change it for you, or we'll make a new query special for you. But I'll just show you what we do. So let's say that I don't want to search for this database identifier. So, okay, let me go back. On the left side, you have a model browser, which is sort of like a tree-like structure showing the data model. And this is where you select what you want your output and what you want your constraints to be. And so anytime you select something, for example, I wanted the gene, I wanted the ID for the gene to show in my output, then I can click on it and it will show as a box over here, a light blue rectangle with the name of the column here. And then I also there's also a constraint here. It says this constraint is that we're doing a query where the ID has to be this number. But let's say I don't want that. I can, you could have changed it just in the query template, but I want to do something different. I want to get rid of this altogether, remove this constraint. See, I just click the X next to that and it's gone. So there's no constraints now. If you tried to run this, it would probably complain because it will say you don't have any constraints. But what I want to do is I want to, let's see, I wrote down what I want to do. I want to do this query for all of the Bombus and patiens genes. And so I can go over here to the model browser on the left side, and I can scroll down until I see organism. Next to organism, if I click this plus sign, it opens up. And then these are all the different kinds of attributes available with the organism. One of them is called short name. That's the genus abbreviation plus species. So if I just click constrain here, a little menu opens up, and then I can select which species I want. And I want it to be Bombus and patiens. And then click add to query. So if you look on the right side now, you can see there's something new. It says short name and then equals Bombus and patiens. So that's a new constraint. So this short name, it's not going to be printed out in the output because there's not a blue box around it, but it will be constrained. If I want that to be printed out, then I can add that. So I can say next to short name, I can click show. And so now short name is in a light blue box. That means it will be printed out as one of the columns in the output. And so now if you scroll down, you can see the columns that will be output. And these are actually here in order according to the way they were added, built in the query builder. And so the short name is last because it's the last thing that I added. But let's say that I wanted that to be the second thing that shows, I can just move the box and it will change the order. And so if I just click show results, this will take a little bit longer. Oh, that's pretty fast. So this shows all the KEGG pathways for all of the Bombus and patiens genes. So you'll notice that, you know, there's less than 4,000 rows, and that's because only a small number of Bombus and patiens genes have KEGG annotation, and that's pretty typical for all the species. Let's see, what else do I want to show? Okay, I'm going to show you something else here on how you can manage your table. Let's say I decided that I want descriptions to go with my genes because I don't know what these genes are. I only have their pathways. So instead of using the query builder this time, I'm going to use the column manager. I click manage columns. And this is just kind of an alternative way of getting different information in your table. So when you click manage columns, you have a list of columns that are already showing, and you can remove any one of them. So let's say I'll just get rid of organism. I changed my mind, I don't want that. I can just click that and it's gone. But what I really want to do is add a column. So I click add column, and that opens up, this is another kind of tree-like view of the data model. It just looks a little different. And so what this shows is in gray, with kind of this light gray highlighting, it shows everything that's already selected. So if you scroll down, you can see some pathway information is selected. And then also, you know, we have our gene ID and our gene source. So I want to click on description. I want to have that as one of my columns. So I can just click here and say add one new column, and then there it is. But let's say I want to rearrange my table. I want to move this column so it's right next to the gene ID. So I can just do that. And there it is. And then I can say apply changes. And then now the gene descriptions have been added. Something else you can do is you can sort these columns. So if I want to sort by gene description, I can just click here, and then it's sorted.
You just have a question, Chris?
Yes.
Is there a maximum amount of output that will result in an output exceeded or other type of error message?
It's really, there's not a maximum amount of output, although if it's a huge query, like let's say you didn't have any constraints and you wanted to do something with all the genes of all the species and in the entire HymenopteraMine, then it might actually, if the query takes, if the Postgres query backend takes too long, you might have a timeout and then get an error. So you can let us know if you have a query that's taking so long that you get that error, and we could help you maybe make it work. But as for the number of rows of output, you can get thousands or 100,000. And it will be, you have your choice of rows per page. So of course, if you have too many rows per page, the more you have, the longer it will take to show up on your browser. But normally I just leave it at the smallest so I can see what it looks like. And then what you can do is that you can export it using this button here. And then, and this allows you to choose which column, if you don't want all the column exported, you can turn some off. You can decide if you want it gzipped. You can add column headers like this. You can look at a preview. The preview is kind of ugly looking because it's all squished together, but basically it's a tab delimited file. So I'm not going to download it now, but that's how you would download an entire table at one time. And then I'll show you later, but I can just show you right now, is that if I wanted to save a list, let's say I wanted to save a list of all the pathways or anything, I can click Save as List here. And then these are the genes. So there's actually only 2,500 genes in this table because that's how many have pathways. And then, or I could save the pathways if I highlight here. So I'm not going to do that now because I'm going to show you that in a different example. The one thing I will show you now is one other thing in these management buttons is this manage relationships. So like I showed you, there's only 2,500 different genes in this because if you say save as list, that's how many show up here. Some of the genes are listed more than once because they have more than one pathway. But when I download it, I want all my genes because I want all the gene descriptions. And so I want those even if they don't have pathways. So what I can do is I can click Manage Relationships, and I can just click Optional next to the pathway things. So those are optional. And if I say Apply Changes, then you'll see that there's 27,000 rows. And if I say Save as List, there's 27,000 genes. And so you'll also notice that you don't have multiple rows per pathway now. Instead, the pathway information became embedded as little boxes. Like some have 0 pathways, some 1, but here's an example where there's a gene that has 2 pathways. So if I click on that, you'll see it opened up. So now I'm going to just go ahead and close this. So when you click Export to download this, it will turn into a normal tab delimited file where you have all the rows and all the pathways, and the missing information will just be empty quotes in your table. Okay, so I have some more advanced things I want to have time to show you. So I'm going to move ahead now to show you how to upload lists of IDs, because this uploading lists is the more common thing that you would probably be doing. So I'm going to click Lists. And when you first click the list tab, you would either get to this upload page or you could get to a view page. So if you're at the wrong one, you want to upload, then make sure you can, up here on the upper left, there's a way that you can switch back and forth. So I want to upload a list, and I'll show you the view page later. So I have some data here that, let me just show you where I got it. So I have some data that I got from a supplemental data set from this paper. And what I'm going to do is use their actual data to do a gene ontology enrichment of differentially expressed genes. Oops, let's see, it's this one. Okay, so this is the supplemental data set that came with this paper, and they were nice enough to provide the background gene list. So I really appreciate when people include their background gene list in their supplemental data, because that allows you to use it for enrichment. So I have this the entire column is highlighted, including the header. So I'm lazy. I'm going to just do that. And then I'm going to copy paste that into this box. And so I hadn't selected type yet. You can select many different types of identifiers, but these are genes. So I'm going to leave it at gene. And then this is Apis mellifera, so I'll choose Apis mellifera. So all the gene IDs are unique, so technically I wouldn't have to select a species, but the query runs faster. Now look at this. This is the bottom of my list. This is a big list, and the bottom of it, it says character 0, and I think that was a mistake. There is an error at the end of the list, but I'm going to leave it here so I can show you that it's okay to even have errors in your list. So the most important thing is that you just have a column of IDs. I'll click Create List. And so notice, I want to remind you that I'm logged in. And that's what's allowing me to be able to do this and be able to save a list. So what happened is that the database first performed a lookup to see if all your IDs are found. And so what it did is it says that I entered 9180 identifiers, but it found only 9178 genes. So if I scroll down to the bottom, it will show me what it didn't find. So it didn't find this error that was at the end of the list, and it didn't find the column heading. But just to point out that if people have typos in their list where they, I don't know, every so often when data is, people create data sets, there might be a mistake. But that doesn't prevent you from being able to do your work. And it also tells you what it found. So I would like to save the list, and I'm going to give it a name so that by default it will just assign, sort of like tell you the time and that you made the list. But I want to change it to something more informative there. And then now it's not going to be saved in my account until I click Save a List of 9178 genes. And there, now I saved it. So the first thing it does when you save a list of IDs is give you some default information about those IDs. And so, and sometimes there won't be any information. For example, our genes don't have secondary identifiers. Some of the genes don't have gene names, but they have gene symbols. Some do. So if you scroll down, you'll see that when you enter a gene list, it automatically enrichment is performed. Although this was meant to be our background gene list, so I'm going to totally ignore what this enrichment says. And I'm going to go and repeat the process for uploading the differentially expressed genes. So before that, let me just click View. Because when I click View, you can see that there is the gene list that I just made. I'm going to click Upload again, though, so that I can upload the differentially expressed genes. And let's see. These, I didn't want to copy the whole column here because there's a bunch of them that just say NA, so I'll just enter these. And so there, that was 533 IDs, and all of those were found. And I'll save that list. And so this is the list where I actually want to do gene ontology enrichment. So now if I scroll down, I'll pay attention to these enrichment terms, which is, so there's something here that you should read every time you do an enrichment. And this is a reminder of things that you should do to make sure you don't have false positives. So 2 important things. First of all, make sure that your list only has genes from only one gene set. And the way that you can check for that is in the table where it says gene source. If you click on the View Column Summary, it will say there's only one gene source, ref-seq. So that's good. Because if you mix genes from different gene sets, that kind of messes up the computations for the enrichment. The other thing is you need to choose a background gene set. So by default, all the gene ontology terms for your species will be used as the background, but that would not be correct because many of the species have more than one gene set. So it should only be one gene set or your particular background gene set. So here, if I click Change under Background Population, there's a pull-down menu that has all of the species genes, gene sets listed kind of there as a default, but there's also the genes that you uploaded. So the background genes that I just uploaded are here, so I can click on this and say I want to use that as my background. And so there, it doesn't look like it changed that much, but sometimes it makes a difference on your enrichment. So it's important to do. And then you can also change the test correction, and of course you can change your ontology. And then here we also have pathway enrichment. And I just want to remind you that for pathways, this would not work for official gene sets. It only works for KEGG. And so again, separately, you would need to change your background gene set. And then this test correction, the Benjamini one tends to be less, not as strict as the other one. So sometimes you'll get additional terms if you change that test correction. You can change your p-value if you want to. You can download these, the results by clicking download, which I'm going to just show you what the results look like here. So this is, here is the, what you would get. This is one of the, this is a gene ontology downloaded. And so you get, this is actually a tab delimited file that I'm showing you as a text file, but this includes the typical things that you would want to include in your supplemental data or the information you'd need if you're going to make some kind of figure. And so, you know, it will tell you the term, the ID, the corrected p-value, and all the genes that are within that category. Okay, so now, so that was doing the list upload.
Oh,
so let me just,
we have another question?
Yes.
If I have gene symbols, for example, LOC, where can I find the equivalent gene IDs like the one just used for the list example?
You can, so this, the list upload allows you to upload gene symbols. So, and then, too bad I don't have that as an example. Oh, actually, if you upload a list of gene symbols, which unfortunately I didn't do, so I don't have an example, but you could then use this, you save the list, you'd have your list saved, and then you can click on this template symbol to gene ID. And what you would do is you click this little box that says constrained to be in, and then you would pick your list of symbols, which I don't have one because I didn't, unfortunately, I didn't save one already to use in the example. So, but anyways, you would, as long as you save a list of symbols, which you can do without upload, then you can use that list in a search with one of these query templates. And also in this one, this particular one, you'd also need to make sure you select your species. So this, for example, this query actually wouldn't work because I constrained it to be in Adocephalotes, but I said the species is Apis mellifera. So sometimes you have to be careful and make sure that you have things set correctly.
And we have another question. Are there areas of HymenopteraMine with little exercises we can use to practice on our own? If yes, exactly where?
So the exercises, the current exercises that we have are in that chapter that I showed on the home page. So if you go, I'll just click here for a minute. So if you go down to the kind of closer to the bottom of the home page, it talks about this chapter. And then if you download this preprint, it has all the data and it has all the figures that go along with the examples, but it's very, very detailed examples. They're probably even more detailed than what I said today. They're very detailed. We'll work on, so what I plan to do actually is the examples that I'm showing you to actually create, make these data sets available. So in the next few days, I'll be adding a tab up here that will be called examples, and you'll be able to get the data that I'm showing you right now so that you can do that. You can watch the video on YouTube and work along. Okay, so I'm going back again, and I'm still logged in. And the next thing I want to do is a region search. So the region search allows you to upload a list of genomic regions. And so this, I'm sorry, I'm showing, this is a very Apis mellifera centric talk, and I know that we have a lot of people working on other things, but those data sets for me happen to be the easiest to find for examples. So I'm going to upload a list of Apis mellifera coordinates, and I want to find genes within those regions. And so the coordinates are in the format of chromosome ID, start, and end. And then they can, there's actually a few different formats that it accepts, but it has to have these 3 pieces of information. And so the most common format would be tab delimited. And so this, and then it allows you to just paste in, if you have these columns in a spreadsheet, you can just paste them. And so, let's see. One thing that I want to point out also, if you have SNPs, which are just, usually at just one coordinate, then you still have to have a start and end. And in that case, those would be the same number. So I'm going to copy these into the box. And you'll notice that these IDs, I'm going to show you later how to find out what should your chromosome IDs be, because of course we know that no matter what database you go to, there'll be a different format. And so I'll show you that in a few minutes. But we actually try to follow now what's available in the NCBI gene assembly report. So the current APIS mellifera assembly had IDs with LG numbers, like linkage group. So, so here we've pasted these in, and if you wanted to, you could extend your regions. For example, you might do that if these were SNPs, so that you can enter 50 KB or something here. But I don't want to, because these are actually regions. They're not just SNPs. So I'll just click search. And so this search went really fast because I had already done that and it was cached. But it's, it might take up to 30 seconds. It's pretty fast in HymenopteraMine because the genomes aren't huge. So so what you'll find in this is sort of an intermediate result page where it will give you results for each region separately. It allows you to download information from each region in various formats or create a list for each region separately. And you'll see that it gives a summary. It tells you what the gene IDs that it found. I only search for genes, so all the feature types are going to be gene, and then the location. You'll notice that it mixes up both the OGS and the RefSeq gene, so both of those will be found. So what I want to do so that I can further do more analysis is that I want to save all these genes as a list. So I will just click Go right here next to Create List by Feature, and this menu only has gene showing because that's the only thing I search for. I click Go, and then now it's we're actually back into a page that looks just like the list tool. The difference is that it actually gives you a default list name. It didn't allow you to create your own name. Using HymenopteraMine, you can always change your name, the name of your list. So this, you'll notice, is automatically doing a gene ontology enrichment. But there's a problem here because if we go to our gene source, we're going to find out there's two sets of genes mixed up. And so what that does is double the gene ontology terms for, because there's two sets of genes, and you'll not get correct results. So what you would do is you would create new lists for each set separately. So let me go back again, so just to show you. Above the column gene source, there's a little histogram box. If you kind of just mouse lightly, it says View Column Summary. But if you click, it actually brings up this little menu. And so this will always tell you, give you counts of data within that column. And it's especially good for data like gene sources where you have a couple that you want to separate from each other. So here I can click one of these, the OGS, and then if I go down to filter, I can select restrict table to matching rows, click that. And then basically it changed it so now only the OGS are showing. So it still has the same name because I haven't saved it. But if you do View Column Summary, there's only one. So now I can save this list. I go to Save as List. And if, by the way, if I would have forgotten to log in, I say, oh, I want to save a list and I'm not logged in, bummer. I could log in now and it would actually, the page would be saved for me. So if you're not logged in already, you'd have to make sure you're logged in so that you can save a list. I click on gene, then a menu opens up. And so I could say, there. So there's a name for that, create list. And then it says a new list was made and how many genes. So, but now I want to do the other genes. So I can easily just undo my filtering step with this undo button. And there it is. So if you go back now, you have both genes again. Then I can now do the same thing with refseq, and then restrict table to matching rows, and then save that as a list. There. So now you can see that I've saved both of them. You'll see that my gene ontology results are still showing, but these are still the wrong results because they are the results that go with this list that's showing at the top of the page. So in order to correctly do the gene ontology, I have to click View, or just click one of the lists, but I'll just click View, and you can see the two lists, and let me click on this, the refseq one. If I click on that, it goes back to this page, but notice now it has the name of the correct list up here. So it's only when you can see the correct gene list that it's actually doing the gene ontology enrichment with that list. And then you can go down and of course you always have to change it. So since this is a region search, I didn't make a separate background gene list. And so what I would do is I would actually select the all the genes. On purpose, I'm going to select the wrong one just so that you can see what happens. I'm going to select Apis florea by mistake. And so if you select the wrong background gene list, it's going to give you this warning. And so the thing you just need to do is click on it again and then scroll down and say, oops, oh, it was the wrong one. And then click the correct one. And there that is. And then, of course, you would do the same thing with your pathways. And then if you're interested in, oh, this one didn't have publications image, but you would have to do that with the publications if you're interested in that too. So, I told you that region search only works if you have the correct chromosome ID. So let me show you how to find out what is the correct chromosome ID. So under these templates, we have a tab called entire gene set. Think of this also as entire genome, not just entire gene set. So it's kind of to like query something for it, like the entire organism at one time. And there's one template called genome assembly to chromosome IDs and lengths. So if you click on that, you can pick your genome assembly. So all the assemblies are here for all the species. You might have to look on the data source page to find out the assembly name for your species, but you can click, check it or select it and then click show results. And then this table comes up that shows all the possible identifiers. And so the column called DB identifier, that's the identifier that's used for the region search. These other things are alternate ways, alternate names. So so for Apis mellifera, it was kind of in the past, the chromosomes were called group something. We also have GenBank IDs and refseq IDs. So and so this, although it might take a little work, because if you have a spreadsheet that has the incorrect IDs, what you might need to do is just add another column, then be able to to change it to the correct identifier. So you'd have to do that work in your spreadsheet before you upload. But at least there's the information here. In the future, we want to work on a region search that would actually be more flexible and allow you to upload any one of those IDs. Okay, so I have one more thing to show you, and I'm running out of time. So I just want to show you something that's really important for the species that we have now is this tab called alias and dbxref. It's not perfect yet. Well, it will probably never be perfect. Nothing ever is, but it's still, we're working toward making it easier for you to get cross-references among gene IDs. And so you go to this alias and dbxref, and then here's I'm going to just click on that first template query where you can enter a gene ID and get the cross-references. And so just to show you an example, I could have used a list that I saved in as my user list, but I'm going to actually select just a different species so that another species could be represented. So here's one of the older ant official gene sets. Then I can, if I say constrained to be in and select that and show results, then you'll see there's all the old IDs. and then the new IDs that go with it. So it will say the gene source of what you entered, and then the cross-reference. And so then if I wanted to actually now do work with those genes, I could then save those cross-references as a list of the RefSeq genes for that. Yeah. And so normally you would do that maybe with a smaller list of genes rather than the entire genome. Let me just show you one more of those. I'll go to the same one. I wanted to show you Nasonia, which has a couple of different gene sets. And Nasonia is, I think, going to actually be further updated. I think there's a recent update. So if so I'm selecting the current gene set, or kind of a later RefSeq gene set. It might not be the very latest now, but I click show results. And then you can see that there's actually a couple of different sources of cross-references. There's the very oldest official gene set, and then there's the EVID gene set. Another thing you'll notice that these counts look pretty low. So there's 10,000 rows of genes, but not, you don't have 10,000 of each one of these cross-references. And that's just because when you're mapping genes to an older genome to older genes to a new assembly, a lot of times genes will get lost, and then you also just have disagreements among genes. Okay, so that is all I plan to show you. And before I stop sharing, I'd like to find out if there's any more questions of what I just showed you.